Today I will be showing you how to add post-translational modifications to your predictions in an experimentally derived protein. So for example, in this protein was experimentally determined, and it's on extracting it directly from the RCSB protein data bank. So to get this done, you need to identify the modification in this platform, in this database, then go further to know the position of which the modification is being carried out using ChimaraX. So let's head straight to the point. So first, you need to download this protein. So you come here, download the PDBX format. Then after downloading this file, this protein, so you now open the protein in ChimaraX. So you open the protein in PDB format. So you open your downloaded structure in PDB on Chimera. So when you open Chimera, this is what you will see. So first, what you need to do is, if your display is showing home, I will suggest you go to display. So from display molecule or molecular display, you see everything that is concerning that molecule. So what you now do is to go to sequence. So when you go to sequence, you use in this prediction. Remember, this is an experimentally derived protein structure. So but we want to predict it using AlphaFold 3. So now you scroll down, when you see modified residue, so here is what here is where you identify whether there was a post-translational modification and how did it occur. So here we have the post-translational modification that occurred was a PTR, and the parent chain is tyrosine. So here is the post-translational modification. So what you now do is to go back to ChimaraX, where you've already opened the protein structure which have been done. So now what you now do is to actually identify where PTR is located in the position of PTR modification in this protein sequence. So how do you get that done? You come to select, then go to residue. So find PTR, come down to here. Okay, we have PTR here. So if we click on it, it will highlight as green here. So here we have it as green and white. So when you click on this, it will show you the number. So this is PTR 64. So that's the position of the post-translational modification that it occurred. So we'll now head back to AlphaFold to modify this in the protein sequence. So go to, there is a link here. So we have the Uniprot ID here, the accession number and the rest. So how to get in, you click here and it will take you directly to Uniprot. So when we come to Uniprot, many informations are here, but what we need for this is the protein sequence. So scroll down, or you can just click on sequence, and here is the protein sequence of this protein. So copy protein sequence, then come to AlphaFold 3. Then now we are in AlphaFold 3 where we'll do the prediction. So what we do is we don't want to do DNA, RNA, ligand prediction or ion. What we want is protein prediction. So we'll now paste the protein sequence here. So since since we are going to add the post-translational modification, we'll now click on these three dots here. Then we have the PTMs, that is the post-translational modifications. Click on it. So remember, we had it here at position 64. So we'll now go back to AlphaFold, and this is 60. and this is 70, so this will be 61, 62, 63 and 64. Click on it, scroll down, we have a 64Y, and so the available design is O phosphor L tyrosine. So that is the post translational modification that occurs. So you add it, save it. So what next will we do? Continue with job project and confirm job. So the job will take a while, I don't know how long, but I've done this before, so we can just open the structure. So let's wait a little for it to open. It should complete in just a second. So okay, so after this, I think it hasn't completed, but I run this earlier, so we have the same, this is the same prediction we did initially, so we can open it. So come here and check for your prediction. So this is our prediction with the post translational modification. So this is what AlphaFold 3 can do. So you can compare this structure with the original structure. Mind you, the original structure has other components, but this is having just the post translational modification and the sequence to predict this protein. Thank you for watching and see you in our next video.